Okay, so welcome to the first seminar, a bioinformatics seminar of this year. Um, I'm very glad to have Dr. Rayan Chikhi. He is working on genome assembly and has been very active in this area. So he studied computer science at ENS Brunei and obtained a PhD in 2012. And after postdoc, and he did postdoc at Penn State University in Paul Medvedev's lab. And then after that, he joined French National Center for Scientific Research, CNRS, as a junior researcher. And then he also participated in Bonsai Bioinformatics team and still working, collaborating with them. And in 2019, now he started a sequence bioinformatics research group at the Center of Bioinformatics, Biostatistics, and Integrative Biology of Institute of Pasteur. Now, so he has his own group and working on the sequence-related the projects. It's great honor to have Rayan. Actually, I worked with Rayan in 2007, and a couple of years later also on, at that time, protein surface shape representation, mathematical shape representation, and he did spectacular job in using the 3D Zelnicke descriptors and 2D Zelnicke descriptors. It's a spherical harmonics-based mathematical descriptor, and which is I'm still using. And it's also, he's a kind of a founder of one of my main projects still I'm working on. So, all right, so Rayan, please go ahead. Thank you very much for joining us today.
Thank you very much for the introduction, Desuke. It's a pleasure to talk to your lab today and to everyone who joined. I will start by saying that I very much appreciate the invitation for this talk. And also, despite the fact that it is a remote talk, I'm also appreciating that you guys keep your camera open, because then I have the feeling of talking to some human people and not a list of names. So it's really a pleasure. I will mention also that you should feel free to raise your hand or unmute your microphone to ask a question at any point. I have about 35 slides, and I think the time for this talk is maybe more than half an hour, so you should feel free to interrupt me and ask a question. The talk is about genome assembly in 2021, challenges in reconstructing genomes. And actually, my first slide is a recap of what Daisuke just said. I was an intern in 2007. I recovered a picture that I hope I have the permission to show in this slide, which we were much younger by then. But so yes, the work I did as an intern was my very first work in bioinformatics. And since then, I've been working for 12 years on bioinformatics, so for me, it's a special moment. So Daisuke introduced me to the problem of representation of binding pockets in proteins, and so introduced me about work using Zelnicke descriptors. So what I did was representing pockets by using a 2D projection, and essentially a protein becomes represented as a bitmap picture in the same way that people do ray tracing in video games. So I remember implementing a web tool in 2007. It was not so common back then. And I was super happy because this work led to many papers led by Daisuke at the time. So I think it really contributed to my PhD career, because I could get some papers before I started my PhD. Back then I was a bachelor student. So after this initiation in bioinformatics, which I really liked, I think it was a very productive and a very good environment for me, I moved to staying in bioinformatics and did a PhD in sequence. So I will skip the beginning because Daisuke already talked about all of this in the very nice intro. Nowadays, my research is about combining algorithms and data structures to the DNA sequences operations we can do in bioinformatics. And in particular, one of them is genome assembly that I will talk about today. So I worked a lot on creating bioinformatic tools for doing genome assembly and working with a class of methods using k-mers, which is becoming increasingly popular in the sequence bioinformatics world. all the pictures that you can see in these slides correspond to organisms which I have closely or distantly contributed to creating the genomes of. So worked on assembling the giraffe genome at Penn State, gorilla genome, goat, okapi genome, and some bacteria on the left. All right, feel free to interrupt at any point during the talk, of course. And I will start with a very general DNA sequencing slide because I actually do not know what is the background of the people who attend this talk. So I will assume that if you do proteins, maybe you have heard of DNA sequencing, maybe you also know well how it works, but if you are more in computer science, this is maybe more distant. So in order to understand the rest of this talk, I will dedicate 1 minute to explain this process. So in DNA, if you look at it very closely, inside your chromosome, you can see a succession of molecules which correspond to letters here. The overall goal of this process is to reconstruct what is the sequence of DNA nucleotides for a genome of an organism. In this picture, we are looking at a bacterial culture. Can you see my pointer, or can you not?
Yes, I can see it.
Oh, you can see my pointer.
Yes.
Excellent, thank you. So imagine you want to know what is the genome of a bacteria. One way to proceed is to make it into culture, to make more of it, and extract the DNA of it, which is inside a tube, feed this tube through a process. The process corresponds in 2 steps in preparing the DNA and then sequencing it into a sequencing machine. The machine is responsible for outputting a set of strings, which are substrings of the genome of interest. And we'll see that in the assembly process, the goal is to reconstruct the original genome given the output of the sequencer. It's tricky because we don't know what is the location of each of the substrings inside the original genome. So it's like a big puzzle. And this will be the topic of my talk. This picture is more or less accurate in how it is done today, but there are many technologies for doing this sequencing step. It's usually a big instrument, but recently, in the last 5 years or so, there has been an advance, which I think is very exciting, but I'm not the only one who thinks it's exciting. It's a technology that is now portable for doing DNA sequencing. It looks like a USB drive. You feed the DNA sample into it, and the way it works inside it is that the DNA is put into a pore, and then you can directly read the nucleotides as the DNA is fed into the pore. The process is not perfect, but it has many advantages, such as portability and also ability to read longer fragments of DNA. So this is just to hint that technology is driving the field in the analysis that we are doing, pretty much the same as I believe proteins are benefited from, I think it's cryo-EM and other technologies for getting better structures. Now we're getting better data in the DNA sequence world as well, which enables many things, and I'll talk about that next. So from a very abstract representation, genome assembly consists of taking a genome that you do not know, getting sequence reads, which are overlapping subsequence covering the genome redundantly. So imagine each of the lines here corresponds to a short DNA sequence. It's a fragment of the original genome. We call them reads. And the assembly process consists of taking these reads, we do not know where they come from, and obtaining an hypothesis of the original genome. The hypothesis is because it is often impossible to perfectly reconstruct the genome, so we get an approximation that is fragmented, but should be as close as possible as the chromosomes that were present in a sample. And this approximation, we call it a set of quantics. Each quantic is a long DNA sequence. Okay, so that's a very quick introduction for the process. Is it all good so far? If it's not, raise your hand or unmute your mic, and I will be happy to develop. If not, I will just continue. My first question to you, I will try to use some interactive Zoom feature, is ask you if you have ever done genome assembly. If it is the case, please raise your hand as if you wanted to ask a question in Zoom. And then I will see if anyone is familiar with this process. Given some audience I know that some people who are listening to this talk have done some assembly. I see 2 hands being raised. Okay. Maybe, oh, I see 4 hands being raised. So that's actually a lower proportion than I had imagined. So this probably means that for 4 or 5 people, this is pretty much going to be standard material. But for the rest of you, I'm assuming you have not maybe performed this operation. So just for the record, I see 6 hands raised. Thank you, and you can unraise them if you want to. So then, if you have not performed genome assembly, you might wonder what is the goal of it. The immediate goal is to just reconstruct the contents of a genome. Have a long file, have a sequence file with DNA in it, which gives you the sequence of an organism. But by itself, it is interesting, but hard to get biological interpretation from it. What is less known is that genome assembly techniques are also used to reconstruct the genome, the transcriptome, that is RNA. It's pretty much the same techniques. Even though in this talk I will talk about genome assembly, you should think that reconstructing the transcriptome, that is the set of mRNA molecules inside an organism or a certain cell type, pretty much the same. You can construct metagenomes, which correspond to collections of bacterial genomes and viruses inside an environment, or just be interested in a set of genes and reconstruct only the genes. So once you have done this operation, this enables a myriad of biology investigations. Foremost is investigation of the phylogeny of species, determining the evolution of genes, or if you are interested in finding novel insertions inside genomes, maybe these insertions are responsible for some condition or phenotype of interest. Finding SNPs inside organisms which are not so well studied, relies on assembly. And then there are some other cell-free DNA structural variants. Then genomics, which is an increasingly popular keyword in genomics, which consists of analyzing multiple individuals from the same species at the same time. And well, on the right, I'm giving some examples of famous genome assemblies being done. There's mouse, there's tardigrades, which is very tricky genome to assemble, and bacteria, of course, there are so many to discover that assembly methods are used all the time for this. So I guess the takeaway from this slide is that it's a very fundamental operation for doing sequence analysis and obtaining biological results if you don't know what the genome is. It's a very old problem. So as far as biology and bioinformatics is concerned, genome assembly was formulated 40 years ago by Staten, who said that with modern fast sequencing techniques and suitable computer programs, it is now possible to sequence whole genomes without restrictions on maps. So that was essentially the birth of the genome assembly problem. So by informatics standards, this is quite old. For the computer science people in the audience, I think it's worth mentioning a short history of the algorithmic aspects that have been used to model genome assembly. So initially, the first way to approximate genome assembly is to consider the widely known problem called shortest common superstring. So if you have n input strings, one way to model the assembly process is to say, I want to find a minimal length string containing all of my input strings. So you can see in this picture here, your input string could be the red strings, 5 of them, and a possible superstring is the one that is in black. And difficulty of this problem is to find a minimal length one. And in fact, it is an NP-hard problem if you have an arbitrarily large amount of strings as inputs. If the number of strings is fixed, it can be solved in dynamic programming in exponential times the number of strings. Still possible, but expensive. So there are some famous algorithms for solving shortest common superstring. One of the most famous ones is the greedy algorithm, which provides an approximation. There are many reasons why this model for genome assembly is not satisfactory. Foremost a genome is not necessarily the minimal length string of its sequencing. The reason is that genomes are very repeated, and because of repetitions, you might have a region in multiple locations in a genome. This is due to alleles and in the human genome and many other elements that move in the genomes and repetitions in general, low complexity regions that make this modelization, this model of assembly not satisfactory. But initially, that's one way to look at it. Then in the 90s, there has been a simultaneous proposal to model the problem using graphs. And I'm assuming if you have been doing bioinformatics that you have seen at least the term, the Bruijn graph, pop up somewhere in papers. And there's another type of graphs called string graphs. And both of them have been introduced at the same time to perform genome assembly. And let me just quickly mention what it is about. So it is in fact the basis of modern genome assembly. So in order to do this operation, you construct a graph where the nodes correspond to the reads, and the reads is what is given by your sequencer. The nodes can also be k-mers, and k-mers are actually short strings of length k that belong to the reads. So in order to construct a k-mer graph, you have to take the reads, cut them into k-mers which are overlapping, and then this is your nodes, your set of nodes. The edges corresponds to overlaps between the reads or between k-mers. It is a very natural way to model the problem, because if you want to solve a puzzle and you know what are the pieces, then you look for connection between pieces. And here connections are symbolized by sequence overlaps. If your sequence overlap is long enough, you're pretty sure that two pieces are adjacent. Difficulty comes from the fact that sometimes you have overlaps which are artifactual because of repetitions, or because you choose an overlap length which is not long enough. But let's put this aside and look at this sample graph that you see in the middle here. Imagine your reads corresponds to a sequence of 3 letters. You can immediately see that it is possible to reconstruct your genome if you do a certain traversal of this graph. And in principle, one way to solve the assembly problem in theory is to return a path of minimal length that traverses each node of the graph at least once. So that's not exactly a Hamiltonian path. It's a generalized Hamiltonian path in CS terms. And you can see that if you do that, you need to start at GAT, do that loop once, and finish at CA. And that would be a reasonable approximation, first approximation of what the genome could be. All right, is it okay so far? Good. As you can imagine, computer scientists who have been exposed to this problem have been asking a natural question: what is the complexity of genome assembly? And the complexity is not so clear, as you can see from this slide. So at the beginning, there has been a strong argument that genome assembly was linear time solvable. Because it turns out, if you model the problem using the Bruijn graphs, then you can actually solve the formulation I talked about earlier using a Eulerian walk. And in fact, Eulerian walks can be found in polynomial time, in fact, linear time. So in a certain way to frame the problem, genome assembly is linear time solvable. But if you incorporate more information and you don't want to solve exactly the same problem as the one that was proposed in 2001, but you modify the problem, then you end up with an NP-hard formulation. So it's one way to illustrate that a biological problem can be framed in multiple ways, and depending on how you frame it, you don't get the same complexity. And depending on how you frame it, you don't get the same results, biological results in the end. So it's very important how a problem is formulated. Two another and interesting intermediate results, in my opinion, are complexity depends on what your instance looks like. So if your genome has repeats which are counterintuitively longer than the reads, so it seems that you cannot resolve them. But in that case, it turned out that there's a result saying that genome assembly is polynomial in the same model as the one where it was NP-hard before. So that result doesn't make sense, but it's true. Another one is if you repeat this one, the next one is more realistic. So if your repeats are either shorter than your reads or are spanned by spanning, and you look at the figure below and you read the red lines, all span the repeated regions, which corresponds to green and black blocks in the genome. And if your reads are nice like this, genome assembly is polynomial and with a unique solution, meaning that you can perfectly reconstruct your genome if your data is adequate. And by adequate, I mean long enough with respect to the repetition that you have in your genome. The catch is that you don't know how long your repetitions are in your genome until after you assemble it. So at least it gives a very natural intuition that the longer you read, the better job you're gonna do. And this has been nicely formalized in a series of papers that I said below. So all of this is nice in theory, but if you want to do practical stuff, you hit a wall for one major reason is that with short reads, the one produced by Illumina sequencers, which have been immensely popular during the 2010-2020 era, none of the theories apply because of the sole reason that the graph that I talked about earlier is disconnected. It's disconnected because sometimes you might not sequence just a small portion of your chromosome for many chemistry reasons, or just because you haven't sequenced enough. But if you have two components, the theory doesn't cover this case. So then you cannot frame the problem as finding a single path. And that's why assemblers tools that do genome assembly will return quantities which correspond to all the unambiguous paths in the graph and do not attempt to find a single walk inside the graph because it's simply impossible given the data that is present. So that was a picture with short reads, Illumina data. Nowadays, I talked about nanopore data, which gives longer reads, more error, but still longer reads. And in that case, there's a nice paper where theory seems to reach practice. Not quite close there, but I think we are getting there. Either way, the problem is still computationally challenging by itself because for large genomes, for instance, humans, mammals, plants, aquatic animals, it has a lot of data to process. I don't know if it is a lot by proteomics standards, but for us, we have to process hundreds of gigabytes of input data, construct large graphs consisting of billions of nodes. And it takes, as you can imagine, months of CPU times for humans and a lot of RAM and so on. So there's a lot of engineering and data structure results needed in order to make the process efficient. In fact, this is an area in which I worked for a long time. If this kind of story resonates with you, I suggest the reading of this nice article which talks about modeling biological problems in computer science by giving the example of how assembly was modeled. So it's a very unique type of articles which shows how you could teach bioinformatics for computer scientists by bridging the gap between an actual biological problem, which is often hard to make a model of, and computer science theory problems, which are very nicely formulated, but sometimes don't quite model what you want in biology. All right, so now I'll shift a little bit gears and talk about more concrete assembly stuff. One main thing to have in mind is that the human genome is still not perfectly assembled and also still not perfectly assemble-able if you were to sequence it yourself. So to give an idea, in 2001, when the first human genome was released by a large consortium of people taking a lot of efforts, it was very fragmented. Like each band that you see on the screen corresponds to a sequence, to a contig. So the sequence was properly assigned to chromosomes and properly oriented, but still you had gaps. And it was not a single DNA sequence per chromosome, but many of them with gaps in between. Nowadays, the picture is much better because we have better data and also better algorithms. So people, and in particular Adam Philippi, are working very hard to make sure that the human genome is now as complete and as gapless as possible. So that is a picture that you can get in last year with some of the recent data and some very finely tuned tools. You can see it's still not perfect, but we are, as a community, people are getting there. You might ask, why is human genome not complete yet? And one of the reasons is centromeres. It's not the only reason, but centromeres are the middle of chromosomes consisting of very low complexity sequences. We knew it was low complexity, but we didn't know what was the sequence composition of them until it was possible to sequence very long reads that cover completely the center of the centromere, and also flanking regions in order to know where to place them. So in this picture, I'm taking a picture from someone else representing a centromere, and each color corresponds to a DNA nucleotide. So you have 4 different colors. And you can see that the middle section, actually all the top section of this picture, corresponds to repeated motifs, which are very similar. So basically each line is very similar to the next line. So if you have data such that your reads cannot span the whole region you see in this picture, there is no way to reconstruct this part of the chromosome, because essentially you will not be able to elucidate the assembly graph at this region. Only if you have long enough reads, remember what I said about the complexity theorem, only if you have reads spanning this repeat entirely, then it is possible to reconstruct this chromosome. So since you have this pattern for each chromosome, that explains why you have at least more contigs than chromosome in human assemblies. And one might ask, what is the state of assembly nowadays? And there is a very nice talk from Adam Philippi who's exactly answered this question for vertebrate and human genomes. I will give some highlights. So vertebrates have very high repeat content. So, I mean, even more than human genomes. So this makes the task of assembling them even more complicated. Diploidicity is another problem, which I'll talk about in a second. And for humans, the prediction that Adam makes is that it would be possible to fully elucidate the human genome within 2 years. We are not quite there yet. A few more gaps are still present, but it's very close. I'll just mention one thing about an assembly caveat, which I think is important to mention for people who actually use this data in one way or another. For instance, even if you do proteins and you know that you have some genes of interest, the gene might not be perfectly assembled for the following reason. So as soon as you have a diploid organism, you have 2 haplotypes. And so I'm representing 2 examples of haplotypes here with a variation between 2 haplotypes and another variation here. The process of genome assembly usually results in a single consensus sequence here. And in that case, which is absolutely not an unusual case, the consensus is a chimeric consensus between the 2 haplotypes at some positions. So you would want to be one of the 2 haplotypes, but in fact, if the variations are sufficiently far apart, it is not possible to do what we call phasing, which is tell that one variation is necessarily on the same chromosome, one allele is necessarily on the same haplotype as another allele. So you get this kind of camera, and this effect is in particular very visible in the very first human genomes, which were a mosaic of multiple individuals. So in fact, the very first human genome in 2001 corresponded to no one in particular. It had a sequence that was a consensus of multiple people. So you saw that effect a lot. And that's important to take in mind, because then it's not actually what was in the sample. And so what I want to mention is that haplotype separation, being able to reconstruct those 2 haplotypes for a diploid organism at a chromosome level, is extremely recent. There has been papers coming out last month, 2 of them, in fact, in Nature Biotech, which advertise the chromosome scale haplotype of assembly of human genomes. So we'll start to see better better data, but better outputs, but it takes a lot of effort and actually good data in order to get around this issue. I also mentioned that genome assembly in a computational aspect is very complex. It takes something around a PhD for a single person or a team of engineers for 1 or 2 years to create such software. The reason is that there are many steps, and the steps are often not so independent components. To give an idea, I took a genome assembler and I plotted each source code file, and each import for one source code to another source code as a graph. And that's an excerpt of the graph. So imagine having to read this code. It's really, it's really complex. And on top of that, there are heuristics everywhere. So sometimes you need to create heuristics in order to make the result better. So there are good reasons for doing this. And if you do not do this, then it's sometimes hard to clean the data, or it's sometimes hard to get results that actually beat the competition. So I like this quote below because it's a little bit provocative, but in fact, the end result is that in order to make good genome assemblies, given that we have imperfect data, there has been a lot of art put inside the software. It is still somewhat true today, even though we have better data. I cannot say that it is straightforward to create genome assembly software. And in fact, I did my PhD on it. And I did one assembler like everyone else who does a PhD on assembly. It takes a lot of time. And give a short overview of what I think is the genome assembly landscape in 2021 for people who are more on the biology side and want to get concrete vision. So you can either choose short reads or long reads to do sequencing. And nowadays, since long read exists, it makes no sense to do short reads. There are multiple technologies for long reads that you might have heard of. One is from Oxford Nanopore, which I mentioned at the beginning of my talk. And the other one is PacBio. And PacBio released what they call hi-fi, high fidelity, high quality reads, which I believe is the best data type you can do for assembly nowadays. CLR has high error rate, it's harder to manipulate. So in fact, people have been making memes about why the new data type is better than the previous one. I will also mention that from a computational point of view, methods which have been developed for assembling short reads are making a comeback with long reads. So in fact, I talk about the Bruijn graphs, which appear more and more inside long read assembly tools, which are starting to be quite useful. So Fly, this one is going to be released soon. And there are also K-mer and assembly validation, which uses methods from short reads for long reads. I'll talk about the Bruijn graph because I've studied this topic for quite a long time, and I believe it's quite fundamental in sequence bioinformatics. And it's also a mathematical object of independent interest. So the Bruijn graph is a graph which has a parameter k. And in this graph, all the nodes are K-mers, which are sequences of length k, which appear inside your data. Edges are very inflexible. So you have an edge only if you have an exact overlap of length k-1 between two nodes. Making an example here with just two reads and a small variation in between them. So the Bruijn graph for these two reads together with k equal to 3 is this graph. So you start with AGC, which is a k-mer of length 3, and then you connect it to two other k-mers, GCC, because GC is an overlap between them, and GC also because of the overlap. And then that's the whole graph. And you can see from this graph that you can get an assembly consisting of two paths corresponding to exactly the two reads you had at the beginning. So it's immediate that those graphs are useful for assembling short reads because k-mer size is actually close to the length of the reads. And if you have longer reads, and especially with errors in them, it doesn't apply anymore because then it doesn't make sense to cut your reads into k-mers. Still, in the short reads era, the Bruijn graphs were actually very useful for assembling. And here I'm representing what they look like. And you can see why assembly is difficult just by looking at this picture. Because if you have your data, you construct a graph, and then anytime there is a repetition inside the genome, that genome here is a fraction of chromosome 14 from human. just half a megabase fraction, short reads assembled using an assembler called Space, which is state of the art. Every time you have an allele sequence or some sort of repetition inside your genome, you get kind of a hairball, because it's impossible to get a graph that is nicely a single segment. You have ambiguities, and because you don't know how to once you enter a repetition, you don't know with which segment to exit it because of the data. So essentially, graphs look like this, and that was the best possible results you could get from short-read assemblies. Still, it's an elegant way to represent genomes, because then you can immediately see repetitions. Where are the repetitions and so on. It's also very computationally efficient. So I will skip to the advances in data structure box, because I actually worked in this area for several years during and after my PhD. And the question we were asking ourselves is, what is the most efficient way to represent the Bruijn graph in memory? And we were trying to optimize space usage. So how many bits per node of the graph? What was interesting is that the initial methods were very memory hungry. So if you represent it naively by just storing k-mers inside a hash table, you get more than 200 bits per k-mer, which makes sense because you need that to represent it explicitly. Later, there has been a lower bound that said you cannot do better than this. At the end of the game, you cannot beat 22 bits per k-mer. That's the information theoretic lower bound. And then during my PhD, I found a way to actually beat this lower bound by changing the model and say, look, we don't need to actually represent a mathematically exact graph for the purpose of doing genome assembly. We can just, have an approximation. And in fact, this approximation will not be wrong in the location where we actually traverse the graph. So this enabled my group, and myself and collaborators, and then other groups, to have very efficient techniques which enable to perform assembly of much larger genomes. And that was only possible using the Bruijn graphs. Using the other graph formalism, it was not possible to do this trick. So then that's why they were very popular for assembling reads and genome assembly in general. But as I said, they are out of the box inadapted for long reads, because you have one error per 10 bases in long reads, and you can imagine that as soon as you use k-mer size longer than 10, then all of your k-mers are going to have errors in them, and none of them correspond to genomes. So the short explanation is that you cannot use the Bruijn graphs to assemble long reads, and there has been some very clever workarounds to still use them. I will talk about one later. So yes, they have been used in short read state-of-the-art assemblers. Okay, yeah, this is what I just talked about. So how to use the Bruijn graph with long reads. I think it's a very neat computational trick, so I cannot resist mentioning it, even if you are not a specialist in genome assembly. So in an assembler called WT-DVG2, that actually comes from, I believe it comes from China, the main developer, they found the following trick. So first, the reads are binned into, imagine your reads are long and you can cut them into bins, successive bin of 256 base pairs. So that's bin number 1, bin number 2, bin number 3, and bin number 4. So that read would be 1,000 bases long in total. If you have another read and you do the same trick, then you get 4 bins and you can try to do alignment between bins using a classical alignment algorithm called Smith-Waterman. And using alignment, you can determine that bin 2 here is similar to bin 2 here. So I put 2, but I should have put another name. You can also determine that this bin is the same as this one. So in a way, once you have done this, you can pretty much zoom out of your sequence. And instead of considering that your read is of length 1000, it's actually of length 4 in another alphabet. And the alphabet is B1, B2, B3, B4, B5, B6. So if you change your representation and forget about bases, nucleotides, but instead decide to zoom out and do another representation, then it is possible to use the Bruijn graphs pretty much as is. So if you forget about base, you can construct a fuzzy Bruijn graph all the bins, and then the representation looks like this. So B6, B2, B3, which is the last part of R2, has an edge with another kemmer, B2, B3, B4, which is of length 3, corresponding to the beginning of R1. That's just an excerpt of what the graph would look like. If you do that over all your reads and all of your kemmers over the bins, it's possible to assemble a genome. And in fact, this program was very successful and very efficient for doing this. So I think it's a very inventive way to do some algorithms using previously known techniques applied to newer types of data using some sort of reduction that loses some precision but still enables efficient analysis. All right, at this point, how much time do you want the seminar to be? I can stop now or do one of the two next topics.
Yeah, let's go on. So it's good. Well, we still have well 10-15 minutes to go. So.
Okay, thanks. Sounds good. So I will go on, and I think there's like 7 slides to go. I will quickly mention one case study. So I participated in the assembly of the giraffe. So we wanted to reconstruct the giraffe genome. It wasn't done before. And that was an outcome paper in 2016 in which we reconstructed the giraffe genome at Penn State along with the okapi genome, which is the closest relative to the giraffe, except that it has a short neck. So you can imagine that this type of investigation is to determine why the giraffe has a long neck, and this in terms of genetic basis. So one way to do it is you assemble your giraffe genome, you assemble the closest relative, and you compare them, compare the genes inside them. And what collaborators have been doing is exactly that. So they found that there is a collection of 70 genes which have evolved in a way that they are responsible for stature of the giraffe and also the cardiovascular system at the same time. So those genes were found, and that was already a satisfactory result because before that nobody knew what was the genetic basis for the long neck of the giraffe. So in terms of biology, I think it's very satisfactory results. I will just mention the bioinformatics aspect of this project, which was a little bit strange because we had some very strange sequencing data to analyze. And I was the person responsible for analyzing it. So if you are somebody who does bioinformatics, you often have to wrangle with some data that is not exactly what you want. In this case, results look like nothing they should. So we requested some specific knowledge from experts and authors inside a form that some of you may be using if you do bioinformatics. It's called BioStar. It's developed at Penn State, and in there you can ask questions, and then specialists will tell you if your analysis makes sense or if you have a particular problem with the software. So as I guess it is usual in biology investigations, there is a lot of behind the scenes work for making results. And often this behind the scenes is absolutely not written in papers. But you should definitely be prepared to know that if you do a PhD in bioinformatics, there is this aspect of exploratory data analysis that is very much present. I'll stop here for the giraffe, but I will mention a few things about assembly tools in general and how good are they and what is the state of the art in this in nowadays long reads DNA sequencing and genome assembly. So there are a lot of tools for performing assembly. I'm highlighting what I think is the best. There are many others. I wanted to ask a simple question. So assume you have a short genome, a bacteria, for instance, which is 1,000 times smaller than a human genome, and it's not diploid. It doesn't have the issue that I talked about earlier about mosaicism. It's haploid. It has a single chromosome. Can we at least do a good job at assembling them with long reads? So in a way, are we mature enough as a field to do genome assembly of simple things and have perfect results? So there has been for the last, I think 5 years, people who advertise that it is not possible to reconstruct a chromosome as a single contig, which is the output of your assembly. And one aspect for this is the following illustration. So if you construct the Bruijn graph of E. coli, which is a famously a famous bacteria, model organism bacteria, which has a 4 megabase genome, so not too big. If you construct the Bruijn graph of E. coli and you choose a small k value, then it is very fragmented because the E. coli genome still has some repetitions, longer than 50 base pairs. If you increase the k value to 1000, then it's less fragmented, but still not a single node. I mean, by node, I mean, it's not exactly the same node definition as the Bruijn graphs. It's unambiguous paths. So initially, you would want the graph to be a single node, which is the case in K2 equal to 7,000, meaning that your assembly is perfectly resolved as a single path. And the reason why 7,000 works is because the E. coli genome doesn't have repetitions, exact repetitions longer than 7,000 base pairs. But it does have longer repetitions than 1,000 base pairs. So there is some sort of threshold. The threshold has been reached by DNA sequencing. You have reads that are longer than 7,000 base pairs now. So by this logic, it should be possible to get one chromosome as an assembly result. Are we ready as a field to achieve this goal? So I was wondering this question, and I looked at a database of 3,000 bacterial genomes, which have been sequenced not so long ago, I think during the last 5 years, using long-read technology. So each line in this database corresponds to a bacteria. And the interesting metric in this table is the number of contigs you get for each organism. So you would want this number to be 1 or 2, depending on how many chromosomes there are in your species, but you definitely do not want 7 or do not want 4. So actually many bacteria have a single chromosome, so you would want 1 most of the time. So we looked and we saw that only 30% of assemblies, 34% of assemblies are not single contig, which means that something is wrong either with the data or with the software. But we cannot do a good job at performing even the simple cases, which are bacteria. So we looked at why. Why is this the case? So one way to investigate such a question is to do simulated data, in which you control everything. So we took a bacteria, we simulated long reads with it, we assembled it using the state of the art, and the result was 3 contigs. So the way we get 3 contigs, that we made sure that the coverage wasn't so large, so that it was a hard case for the assembler, but still not impossible. So then we see, okay, the result is not what we want. Can we investigate why? And with my PhD student, we actually did this investigation. And the way we did this investigation was going back to the raw assembly graph before the software could do its heuristics. So in this instance, we took the graph. Remember, the nodes of the graphs are the reads and the edges are the overlap. And we colored this graph with the contigs. So we had this purple contig, this red contig, and the green contig. And we noticed that there was a missing connection, for instance, between the red and the green, because simply the assembler did not find enough reads to cover this gap. And here there is a missing connection between the purple and the red contig because there was a big repetition inside the genome. So it is possible to understand why your software doesn't produce the intended results if you go back to an intermediate representation, which is what we did here. And here the intermediate representation is the graph. We could see it because it was a small genome. For a human genome, it's much, much harder to do. It's actually possible, but much harder. So my student, Pierre-Marie Jean, was the software to actually do this investigation. And I would just mention that it's a pipeline. And one good aspect of it is that it enables us to do metagenomics. So if you sequence multiple bacteria at the same time, which is sometimes what you need to do because it's impossible to cultivate, then you end up with a genome assembly, which is not for a single genome, but for many genomes at the same time. And often this is pretty much the same as assembling all of the genomes individually because you can separate them. For instance, this graph here corresponds to one species. This graph corresponds to another species. So you could definitely say the assembly result of this graph is going to be a whole species, and that's it, there's no more. But here, if you have two very close genomes, two strings, you cannot separate them. So it's possible to investigate that here. If you do, for instance, taxonomic assignment on the nodes of the graph, you could say, look, I was expecting 5 genomes, I got only 4. Why? It's because Salmonella has, inside this sample, had 2 genomes which are too close to be separated. So we could do this graphic investigation and understand why the software was not able to produce the intended result, which would have been 5 species. So I will not say much more about this, but I think it's good to investigate in genome assemblies in a certain way. And I will, I will conclude at this point. So the takeaways from my talk is that genome assembly nowadays is still a complex operation done with long reads. It's one of the two fundamental sequence bioinformatics problems, along with genome alignment or reason alignment, which I didn't talk about. And in this subfield, it turns out that data structures and algorithms play a huge role in methods. So that's why, I was very interested in computer science. I am still, because it is extremely useful in this aspect. And also, people have been doing large amounts of Python, C++, and Rust nowadays programming for making tools for analyzing data, because it requires high performance. It's a lot of code. And I'm guessing it's the same proteins, but it's a lot of programming being involved. I mentioned two open problems. One is the strain resolved metagenomics, what you saw earlier about species cannot be separated from assemblies. And a natural open problem nowadays is how to get highest possible quality human assemblies from only long reads. It seems that we are close, but still not quite there yet. Okay, so I will thank you for your attention. I will acknowledge the following people for materials of this talk. I will also acknowledge my group at Institute Pasteur and some funding from some funding sources. And I will leave you with this inspirational Twitter quote that I like when I talk about assembly. Finding your way in life is like finding the genome in the Bruijn graph. It's very easy to find one path, but it's very hard to find the best path. Thank you very much.
Okay, thank you very much, Ryan. Well, it was very interesting to me. I was not very familiar with genome assembly, and I learned that there are lots of still problems and uncertainties in even human genome assembly. Any questions from the audience?
Hello.
Yes. Yes, go ahead.
I actually have a quick question. So, what actually limits us from reading the entire genome? I mean, why do we have short reads and long reads, and why can't we just read the entire genome?
Yeah, so the sequencing technology is a limiting factor. So one limitation is DNA polymerase. That is, I think, a protein that you know better than I do, but it's used inside the sequencer in order to replicate DNA. And apparently this protein kind of gives up after 1,000 or so rounds. So that's one chemical aspect. And also obtaining high molecular weight DNA, high quality long DNA is apparently not so easy. So your sample naturally breaks if you try to clone it in order to sequence it. That's pretty much the state of my knowledge. I'm sure some other sequencing people are more aware of the limitations than I am.
Thank you. I actually had another question too. So, so de Bruijn graphs aren't the only way that de novo assembly is done, right? I mean, you talked about one of these. One of these approaches using Smith-Waterman briefly, but I mean, there are the tools that you listed. Few of those tools use Smith-Waterman to do the de novo assembly too, right? So, what makes? Why is de Bruijn good, and in what cases is Smith-Waterman or other algorithms better?
Yeah, that's a very relevant question to ask. So Fly is a modification of de Bruijn, and for instance, Canoe, HiCanoe use string graph, which I didn't talk about them, but it's basically the same type of graph, except that you don't cut your reads. And then you use a variation of Smith-Waterman in order to get the edges. So it makes a lot of sense that if you don't cut your reads, you are able to elucidate repetitions in a better way. But then constructing the string graph is a challenge because you kind of need to do some sort of all versus all alignment between your reads in order to find the overlaps. And you can imagine an n square with n equal to a few millions is problematic to perform. So there are some computational aspects linked to it. And also string graphs are messy in their own ways because you kind of need to choose a correct minimal overlap length that you tolerate. And if you choose this wrong, you might miss some overlap, or if you might include too many and then you have too many repetitions inside your graph, which makes the thing problematic. So that's my short answer.
Thank you so much.
Welcome.
I have a quick question. What's the typical length of contigs?
Oh yeah, I absolutely didn't talk about metrics for assembly, but depending on your technology, for long reads, for instance, it's not unusual to get contigs of megabases of length. For short reads, it was more usual to get contigs of length 10,000, 100,000 base pairs, not much more.
Thank you.
Welcome.
May I ask a quick question? So, very nice talk. Do you see similarity between this metagenomics type assembly and then multiploidy type assemblies? So many of the genomes, like, I don't know, maize genome, are not diploid, are not haploid, but they have many ploidies. So, and that comes very similar to having, let's say, 2 species of Salmonella and then your kind of thing. Do you see similarity in there? And then do we have any way of dissolving the issues there?
So absolutely there are similarities. And this is being investigated for, I mean, at least in my group, is to use very high quality data and very stringent overlaps, which basically make sure that if you construct a genome, it's only from the same species or same string or the same haplotype. There are also a whole different field that I don't really touch, but I'm aware of. Once you have, imagine you are able to reconstruct only one of the haplotypes. And it doesn't matter if it's precise. It can be a mosaicism of all of your strains or all of your chromosomes for maize, for instance. Then what you can do is align back your reads to this haplotype, do some variant calling, and then you can just get the positions where there are some differences between the haplotype. And then using for instance, a single haplotype as a backbone, it's possible to perform phasing and reconstruct all the other haplotypes. That's a very interesting computational problem on its own.
Thank you.
So hi, Ryan. So technical question for you. So I was originally from the cytoscape team. That's actually we were making software to visualize the big graph. So we always have questions about the divergent graphs, like you know, because it's a very large graph, right? And how actually you guys visualize, or you guys don't want to visualize that, and what actually implementation or like software do you use to store and process this very large graph? I guess you guys probably don't use whatever, like, you know, this what a network X or something like that. It's not quite efficient for super large graphs. You know, yeah.
Oh yeah, this is a very important question for people who manipulate those data. So we, I guess this field has matured to when using a certain graph format, which is called GFA, Graphical Fragment Assembly, I think. It's called GFA. So we store the graph in pretty much in text form.
Text form.
text form with the
Okay.
nodes and the edges very cleanly represented. We don't have a binary format yet. As a field, we are not that mature. And,
Okay.
and then we can load the graph. And nowadays, the software that is being overwhelmingly used for showing assembly graph, for instance, the one I'm showing now, it's called Bandage. It was written by Rayan and Wake, and it supports graph to around 1 million nodes, but not much
I
more.
see. Yeah, but are you going to be able to render the node and the edges, just like drag them?
Yeah, you can drag them.
Okay, cool.
But to be honest, representing more than 1 million nodes, it's still an open problem because it takes a lot of resources.
Yeah. Right. Cool. Thanks.
Okay.
Ryan, I have one biological question and one some technical questions. So if you have problem for diploid genome, like a human genome,
yeah.
Can you comment how reliable the SNPs analysis people are doing currently?
Oh yeah. So for instance, the human genome is a particular one because it has been extremely curated. SNP analysis, you can basically divide into 2 types of regions. So if you are inside a unique region of the genome, by that I mean that your read maps there and nowhere else, possibly, then SNP's accuracy is 99.9% using software such as deep variant, both recall and precision to be complete. So in those regions, it's perfectly fine. But if you are looking for calling SNPs inside extremely complex regions such as centromeres, then it's a whole different story because even mapping reads there is very hard. So until very recently, the VCFI, which corresponds to variation you could get from genomes, were limited to a certain portion of the human genome, which I think was 75 or 80% of the genome. So you had 20% of the genome we don't touch. We are not trying to call SNPs there. Or if we call SNPs there, it's very unreliable.
Sure. Okay. The question I have is, once you have a technical question I have is, once you have this the Bruijn graph, can you output, let's say, with some like a quality score, can you output, I don't know, top 10 best possible plausible assembly?
Yeah, but I mean, we were looking, many groups that were interested in that, and it turns out that it's not as easy to implement as it sounds. The main reason why is because of combinatorial explosion. So you have more than 10 repeats in a graph. So there is really, and also there is no way to prioritize your paths. So because of that, you essentially have millions of possible solutions which are equally likely.
Okay, yeah, thank you.
Yeah,
Yeah, because I have like a very naive idea of applying this as a graph to protein structure, connecting fragments of structures, and I thought, for that I wanted to have top 10 best model, top 100 best models, yeah.
We can talk about it. It really depends on what the data look like.
Yeah. Okay. Any other questions from the audience? Okay. All right. If not, thank you very much, Rayan. Again, it was really great to have you here. Yeah. Thank you. Let's give him another hands to his talk. Thank you very much.
Thank you, Ryan.
Thanks much. My pleasure.
Thank you so much.
Thank you.
All right. Thank you very much for people who participated this time. We have a next seminar in 2 weeks, and the topic is on now in application of AI, well, like machine learning to drug discovery. So which will be probably also very interesting topic. Thank you very much, Ryan, again, and for participation. All right.
Thanks. I look forward to see you in Indiana at some point.
Oh, sure, of course. All right, thank you.
Don't please close the meeting. Leave it open.
Okay, okay, sure.
Awesome. You take care. Goodbye.
Bye. Nice to meet you.
Bye, Ryan.